Hello students, hope you all are doing well. Previously in our classroom lectures, we were discussing about the process of respiration. In the respiration, we were particularly studying about the respiratory substrate, that is the glucose. So far we had studied the process of breakdown of glucose into the pyruvic acid, which was called the glycolysis. And the pyruvic acid will enter into the mitochondria, the matrix of the mitochondria, where it is decarboxylated to a compound that is acetyl-CoA. This acetyl-CoA will undergo a cycle and regenerate it into itself, the acetyl-CoA. That cycle is called Krebs cycle. So we have discussed these two steps, that is glycolysis and Krebs cycle so far. So the product of the glycolysis and Krebs cycle, we get the high energy coenzymes, that are the NADH2 and the FADH2, into the matrix of the mitochondria. So we get high number of NADH and FADH into the matrix of the mitochondria, and still our large amount of our energy is trapped into these high energy coenzymes. So these NADH2 and FADH2 will again oxidize to release the ATPs or the energy trapped within themselves. So the process of oxidizing the high energy coenzymes, that are NADH and FADH, by the series of electron carrier proteins, the proteins which can carry the electron and pass on to the next level, is called the electron transport chain. The location of the electron transport chain will be the inner membrane of the mitochondria. In the inner membrane of mitochondria, we can clearly see this is the inner membrane of mitochondria, also called the cristae. It is not regular. It have incision-like structures, so that the maximum peripheral area can be covered, so that the more number of electron transporters or the ATP producer oxysomes can be produced in the mitochondrial membrane. So the NADH2 and FADH2 will enter from the matrix to the inner membrane of the mitochondria in the series of these electron carriers. These electron carriers are present in complexes, the four complexes: complex 1, complex 2, complex 3 and complex 4. They will release the electrons and protons, and these electrons and protons will move into these electron chains. That's why it is called electron transport chain. As a result of the electron transport chain, the NADH2 and FADH2 oxidizes into protons. This large number of protons will accumulate from the matrix to the intermembrane space of the mitochondria. As a result, after the electron transport chain, many number of protons are accumulated into the intermembrane space of the mitochondria, and we will discuss later the oxidative phosphorylation process. In the oxidative phosphorylation process, these protons will again back to the matrix due to the generation of the concentration gradient, because after the electron transport chain there will be more number of protons in the intermembrane space as compared to the matrix. So they will try again to diffuse into the matrix of the mitochondria. So they will diffuse by the particles oxysomes. Whenever the 3 protons will enter the oxysomes, there will be the generation of 1 ATP so far. Hence, when we will go through the electron transport chain, we will see one NADH2 will move 10 protons into the intermembrane space. One FADH2 will move 6 protons into the intermembrane space, thus creating the almost 3 ATPs, because one ATP is synthesized from 3 protons. So almost 3.3 ATPs are produced by one NADH, and exactly 2 ATPs will be produced by one FADH. So we will see the mechanism of the electron transport chain. So here are the electron carrier proteins that are present in the complexes. Let's talk about the structure of the mitochondria. We know the mitochondria have an outer membrane, the inner membrane which is folded, called the cristae, the intermembrane space in between, and the cytoplasm of the cell accumulate all the cell organelles. Same like the matrix of the mitochondria enclose all the components, all the other components of the mitochondria, like the DNA and the ribosomes. So let's compare with this diagram. So if this is the matrix, so outer to the matrix is the inner membrane which is folded. Outer to the inner membrane will be the intermembrane space, and outer to the intermembrane space there will be the outer membrane and the cytoplasm. So in the inner membrane of the mitochondria, we have discussed the location of the electron transport chain is the inner membrane of mitochondria. In the inner membrane of mitochondria, all the electron carrier proteins through which the electron will be transported with the redox potential gradient, there are many proteins present. In the complex 1, the complex 1 is called NADH oxidoreductase. There are two proteins. One is FMN protein and the another one is iron sulfur proteins. There is another complex which is called complex 2. The complex 2 is slightly embedded in the matrix and slightly embedded in the inner membrane of the mitochondria. It is also called the succinate dehydrogenase, which is a step of Krebs cycle. Whenever there will be the formation of fumarate in the Krebs cycle. Next is complex 3rd, having the cytochrome proteins and the iron sulfur center. There are two cytochrome proteins, one is cytochrome C1 and another is cytochrome B. And in between them the iron sulfur proteins are also present. The complex 4 also have cytochrome proteins, the cytochrome A1 and the cytochrome A3. In the inner membrane of the mitochondria, the cytochrome A3 will be the last electron acceptor. So complex 3 is called cytochrome reductase and the complex 4 is called cytochrome oxidase. In between complex 1 and complex 3 or in between complex 2 and complex 3, there is a mobile carrier protein. Why it is a mobile carrier? Because it can move with their location. The ubiquinin, the UQ mobile carrier, will either accept the electrons or protons from the complex 1. It will move, accept from complex 1 and transport the ingredients to the complex 3. Or either it will accept the contents from the complex 2. It will move to the complex 2 and take the electrons and protons and will donate to the complex 3. So that's why it is a mobile carrier. Same like the ubiquinin, the cytochrome C is also the mobile carrier. It will accept the electrons from the complex 3 and it will donate the electrons to the complex 4. The cytochrome C cannot accept the protons. So in all these complexes, the 4 complexes, there are many proteins. The FMN, FES in the complex 1, cytochrome C1, cytochrome B in the complex 3, cytochrome A1, cytochrome A3 in the complex 4. The complex 3 can accept the electrons or protons from the either complex 1 or complex 2. The electrons and protons movement will not occur between the complex 1 and the complex 2. So they are antagonists to each other. Means the ubiquinin either accept electrons from 1 or either accept from the 2. So in all these proteins, the FMN protein, the FAD protein that is present in the complex 2 and the ubiquinone can accept protons as well as electrons. So they are both electron and proton carriers and they can accept 2 electrons and 2 protons at a time. Meanwhile, the iron sulfur proteins, all the cytochromes and the copper ions that are present in complex 4, cytochrome oxidase, can only accept the electron as well as they can accept only 1 electron at a time.